Okay, good afternoon, everybody. It's a pleasure for us to host today Professor Benjamin Dekel from Sheba Medical Center. I'll say a few words about Benny, and then he can start. So Professor Dekel is the director of the Pediatric Nephrology Department and director of the Pediatric Stem Cell Research Institute at Sheba Medical Center. He's a professor of pediatrics, human genetics, and biochemistry at Tel Aviv University. He also serves as the president of the Israeli Stem Cell Society. He received his BSc and MD degrees from the Technion, and I had the pleasure of being a classmate with Benny during his studies. We had a lot of fun together. But after that, he left Haifa, unfortunately, went to do a PhD in Weizmann Institute and a postdoc in Weizmann Institute. And he had his training in Sheba and Schneider Institute in pediatric nephrology. Benny's research focuses on the kidney in the field of human renal stem cell biology, cell therapy, and regenerative medicine, with application also in pediatric renal cancer. He's really one of the pioneers and leaders in this field in the world. He identified unique progenitors in the kidney, and he's using them both as sources for regeneration therapies in the kidney, as well as he identified their role in cancer, and he's targeting them to treat cancer. Benny is an elected member of the American Society of Clinical Investigation. He won several awards, among them an important award that we give here in Rambam, the Yudim Prize for Excellence in Cancer Research, and the Israel Medical Association for Outstanding Medical Scientific Contribution. And I think today he will continue to teach us about his work. And Benny, please, just a short comment. If anybody has a question, please use the Q&A possibility at the bottom of your screen, and we'll use these questions and deliver them at the end of the talk. Benny, bevakasha.
Yeah, so thank you for the kind words. We studied together, and I would get a 95, and Lior would get a 96, and that was always frustrating, but also we kept in close connections over the years. And I need to share my screen. Just a minute. Here it is, the Rambam. Just a minute. So again, thank you, Lior. The name of the talk is Taking the Kidney to a New Dimension, and I'm going to discuss some new results in the lab. And this won't be only regenerative medicine, but other topics. And of course, the clinical problem we are dealing with is chronic kidney disease. I'll tell you a bit about it. I'll tell you about the need for human kidney forming stem cells. We'll discuss the natural processes of kidney growth that actually dictate these kidney forming stem cell characteristics. Describe some types of kidney forming stem cells, from the fetal, the kidney pluripotent stem cells, and adult kidney. I'm going to show you how the move to a new dimension to 3D culturing has gained us potency and other aspects. And we'll discuss during the lecture some regenerative medicine aspects, bioengineering, and of course, disease modeling. So let's start with a patient. I'm a pediatric nephrologist, so we see a lot of renal malformations, kidneys that are born with malformed kidneys, and this is a malformed kidney. This is a dysplastic kidney. You can appreciate the renal parenchyma is hyperechogenic. Nephrons have not been formed well. And this kid will succumb to chronic kidney disease. Of course, chronic kidney disease is a huge epidemic. It's not only renal malformations, it's hypertension, diabetic nephropathy, and so forth. But the principles are sort of the same. You lose nephrons, you lose nephron cells, and we stage chronic kidney disease by implementing glomerular filtration rate, renal function, or by drawing blood from the patient and analyzing creatinine levels. And we put these parameters into formulas, and we can really tell you what stage are you on in your kidney disease. And we usually see, the patients follow these paths towards end-stage renal disease. End-stage renal disease is when you have 15% of your function, at least less than 15% of your function. And here you need to call the transplant surgeon or the dialysis service, because you need to put the patient or the kid in dialysis. And of course, it's a huge clinical problem. No specific treatments. Now there are at least some, the SGLT inhibitors. I'm not going to discuss these, but as a clinician, this is a frustrating place to be in when you have patients and you don't have too much or nothing to offer them. And this is, of course, the paradigm you all know. Current therapies for end-stage kidney disease, transplantation, one of the major medical advances over the past 60 years. And better, this is better off dialysis as 4 times a week, 4 hours, and so forth. And these options are something we need to defeat or come with better solutions prior to reaching end-stage kidney disease. So basically, the paradigm here in my talk is what we call the kidney repair shop. We need to grow nephrons, nephron parts, nephron cells, and for that, we need the optimal kidney forming cell or kidney forming stem cell. And let's discuss what would be the characteristic of an optimal kidney forming stem cell. So it should be expandable, right? In culture, long-lived, self-renewing over many generations, ex vivo culturing. Should maintain renal potential. I mean, what's the relevance if it doesn't have a renal potential to generate renal tissue? Preferably multipotent. That is, to give rise to the different cell types of the nephron. That's another characteristic. Should be transplantable, right? We want to, just like we do with bone marrow stem cells, we want to engraft these kidney-forming stem cells efficiently and generate healthy tissue in disease models and in patients, of course. So this is transplantable. And let's not forget primum non nocere. This should be a safe procedure, safe cells, no tumorigenicity, and so forth. So this is the definition of an optimal kidney forming stem cell. Now, to understand if such cell exists, we need to learn about the natural processes, as I said. Kidney development, and this is the basis of understanding kidney forming stem cells. But before we go into kidney development, I really want to share with you the sources that would entitle us with kidney-forming stem cells, kidney-forming stem progenitor cells. This would be the fetal kidney, as I said, one option, the adult kidney, and here I draw the line because here the cells would be ready to use, and we need to culture them, we need to cultivate them from the adult kidney, from the human fetal kidney, and expand them outside of their body. Pluripotent stem cells, of course, we can start off with the pluripotent stem cells, whether it be an IPS, a human embryonic stem cell, but here it's not a ready-to-use cells. We would need to differentiate it into the renal lineage and then expand it and use it for our regenerative purposes or disease modeling. I'll show you later. And of course, reprogramming. Take a different cell type, not a renal lineage cell type, a non-renal lineage cell type, shove in transcription factors of the renal lineage and transform it into a renal forming stem cells. So these are the options. Now, Willie, you have to remember that mesenchymal stem cells and cord blood stem cells and fat stem cells and brain stem cells and bone marrow stem cells are not kidney forming. They're stem cells and they could be cultivated ex vivo, but they're not kidney forming stem cells. So we're in the party that does kidney for kidney. We don't do, for kidney tissue, you do kidney forming stem cells. And we'll start with the human embryonic kidney because we will discuss human kidney development, and this will tell you what are the options to derive human kidney forming stem cells from a developing human kidney. And I'm going to take you back to the fifth week of human gestation, where a branch of the Wolvian duct invades the metanephric mesenchyme, which is part of the intermediate mesoderm. And then you have mesenchymal stem cells, and you have ureteric bud epithelia. This is ureteric bud epithelia, are continuation of this Wolfian duct. Now, these metanephric mesenchymal cells will give all epithelial cell types of the nephron. Okay, so they give rise to the nephron. These guys, the ureteric bud epithelia, undergo branching morphogenesis to give rise to the collecting system. So the one that would drain the urine, and not only collecting ducts, but also part of the renal pelvis, the ureter, and even part of the bladder. So this is the ureteric bud epithelia, and these are the metanephric mesenchymal cells. And of course, they have signals that cross-signal to each other. Ureteric bud epithelia signaling, this generates the niche for the mesenchymal to epithelial conversion to generate a nephron, and the mesenchymal cells giving signal to the ureteric bud to branch and give rise to the collecting system. Now, this gaining your nephrons, and remember that, starts from the 5th week of human gestation. This process goes on to the 34 weeks of human gestation. And after that, you do not generate whole nephrons, and there's a stop. The metanephric mesenchymal cells actually go away, and you don't see the nephrogenic cortex, these very delicate cells, and you stop gaining nephrons. And this is how it looks. This is a section. These are the ureteric. This is the ureteric bud. This is the condensate. These are the metanephric mesenchymal cells. And here they're differentiating to these early nephron figures. This is a pretubular aggregate, and you know, they start generating these S-shaped and comma-shaped figures. These will be nephrons. These will be nephrons that will elongate and be nephrons, and these will branch again to generate the collecting system. So this is the process of gaining your nephron, and the metanephris is the site of nephron production, whole nephrons. Some molecular biology of how this happens. The many fantastic studies from various groups on how this happens. Against the ureteric bud invades the loose metanephric mesenchymal cells, these cross signals on Wnt9b, GDNF, and then once the metanephric mesenchymal cells aggregate on the ureteric bud, they generate the cap mesenchyme. These are the condensates. These are self-renewing cells. They express a variety of renal transcription factor. Now, this is crucial. These transcription factors, these renal lineage transcription factors are crucial because if you want to sort out these cells from the human fetal kidney, we need to show that these guys are overexpressed by any analysis, by RQ and so forth. And this tells you that we have the right cells in the culture dish. Now, once these cells start differentiate, again, they generate a pretubular aggregate. A Wnt4 signal comes up that pushes them further on. E-cadherin starts to be upregulated. They're vimentin plus, and here they're E-cad plus. So this is a mesenchymal to epithelial conversion. Okay, and then we described limitation in Wnt4 many years ago that precludes these pretubular aggregates to go further into comma-shaped and S-shaped bodies and to the whole nephron. Now, interestingly, once the whole nephron is formed, the renal embryonic transcription factors go away. Well, at least WT1 stays in the podocyte. There are some remnants, but a measure to reignite nephrogenesis, what might be by the introduction of these renal transcription factors in adult life. So this is the process, which was beautifully drawn by Oren in the lab, of nephrogenesis and differentiation of the embryonic renal stem progenitor cell pool. Now, these are self-renewing cells, and they're multipotent because they can give rise to the entire nephron cell types. 6-2 is the transcription factor that is a hallmark of their presence, and it really also gives them some form of self-renewal during the 5 to 34 weeks of whole nephron production. So this is how it happens. This is the whole nephron, and these guys go away. Now, this is embryonic life. Now, what happens in the adult? Now, remember, you're born with a kidney of about 4 centimeters, and you end up with a kidney of 12 centimeters, okay? So there is postnatal growth, but I want you to understand that these are not whole nephrons being generated. Mostly the nephron elongates and gives rise to and the kidney is larger in dimension. Now, in physiology, the fish, for instance, can generate whole nephrons in adult life. It has the remnants of these embryonic renal progenitors in adult life. They have a niche for these stem cells, the embryonic stem cells, and they can generate whole nephrons. And here, as opposed to fish, we mammals, we lose nephrons and we adapt by enlarging them. Okay, we adapt by enlarging them, and I'll show you other mechanisms in the next slide. But the fish, for instance, has a nephrogenic response. He has these ready-to-use nephron progenitors, again, embryonic life, to generate whole nephrons. But we start up with 1 million nephrons, and we eat too much, and we have high hypertension and diabetes mellitus and Western way of life, and we lose nephrons and nephron cell types. So adult nephrogenesis, mammal no, fish yes. Now, more characteristics of adult kidney growth, and I take you back to work we did a couple of years ago when we used multicolored mice, the rainbow animals, mice, to study adult kidney growth. And here you can really fix a gene color in a specific cell and follow clonal evolution in any organ. It's a really neat tool. And when we did it, we were the first to show that there is a continuous overturn of cells in the adult kidney. There is a cell turnover, but it's not by generating whole nephrons within each segment. This is the proximal tubule, and this is the loop of Henle, and this is this distal, and these are collecting ducts. There are clonal expansion of lineage-restricted progenitors, meaning that each segment is responsible for its own destiny, or I call these local cops, okay? There is processes of local regeneration within nephron epithelia. These are adult cells that go into clonal divisions and generate clonal expansion that are lineage restricted, are only within the proximal tube, only within the loop, distal, collecting, and so forth. You can find bipotency in the collecting system between 2 cell types. I'm not going to go into it, but basically these are lineage restricted. And when you have a Wnt signal, when you generate a Wnt signal in each of these clones, you can get larger clones. So you can envision a Wnt responsive cell in each of these segments, Wnt responsive cells that are amenable to Wnt induction and clonal expansion. Again, adult cells. Now, remember that we shed around 70,000 epithelial cells per hour into the urine. So of course, it's only natural we regrow locally, local regenerative processes in the adult nephron to combat this urine loss. And of course, we would lose our kidneys into the urine if this mechanism wouldn't have existed. So this is how we locally regenerate the adult kidney as opposed to whole nephrons during embryonic life. And this is some examples of the clones, which are always always are they're unicolored, and they are lineage restricted within specific segments of the kidney. So two systems exist, okay? I want you to understand this. Early in development and neonephrogenesis, there is a system whereby multipotent stem cells, self-renewing 6-2 plus, generate whole nephrons, whereas opposed to a late system B, late development after the nephron has, after renal epithelium, nephron epithelium have been generated, and it's in late development and throughout adult maintenance and regeneration, where a specific cell type within a specific segment give rise to its own. Okay, so this is pre-met, mesenchymal to epithelial transition. This is post-met, unipotent progenitors, as opposed to multipotent stem cells. Again, you know, it's usually a state. These cells go into a state that, and they behave as unipotent progenitors. And this is local regeneration of kidney segments. So there's lineage restriction moving from fetal life to adult life. And I like to draw this like this here. The multipotent stem cells were you know, they're the stem giving rise to whole nephrons, and these are the unipotent progenitors in the adult kidney, which are responsible to their own sub-branch. And again, some more examples. This is the fetal kidney, which I like to see it just like the hematopoietic system. It's a mountain where you have undifferentiated, more differentiated, more differentiated. There's a hierarchy, just like hematopoietic stem cells, and therefore this human fetal kidney, to get the progenitors, are amenable for cell sorting. You can sort out the undifferentiated cells, whereas the human adult kidney, it's much less amenable to cell sorting, because it's a plateau where, you know, cells are acting as, so much more difficult to sort out specific cell types. You need to find other ways to grow these progenitors, and I'm going to show you that later. And this is again old data where we could get the undifferentiated cells, you know, the tip of the iceberg. With NCAM1, we discovered a biomarker that is relevant to human fetal kidney progenitor. You see very nice expression in the cat mesenchyme cells. It's it co-localizes with 6-2, and this is Orit's work, Steinberg many years ago, where we could get out these we can sort out, getting the human fetal kidney, do the NCAM1 sort, and get the stem cells in the Petri dish, do the in vivo assays to regenerate and get tubular structures, get kidney tissue forming and disease models. This was published in 2013, and it was really relevant. The identification of these cells were relevant to what we call Wilms tumor stem cells, because this informed the Wilms tumor stem cells. So it wasn't only getting cells for regeneration, it was discovering cells that are malignant cancer stem cells that could be targeted with immunotherapy. So this is the paradigm of human kidney stem progenitor cell identification from the human fetal kidney, using the hierarchy we have in the human fetal kidney and sorting these progenitors with NCAM1. Now, we did, you know, a comprehensive analysis, and basically we can prospectively isolate not only the very early phases of the undifferentiated NCAM 6-2 cells, but we can also isolate further on, you know, during the mesenchymal to epithelial transition phases. We can get the renal vesicle cells, the S-shaped body, and even mature tubules by analyzing three markers: NCAM, CD133, and EpCAM. These are three players where, just like the hematopoietic system, where you have SCOP1 plus or CD34 plus, or you can use by positive and negative sorting, you can really fish out the renal developmental progenitors from a specific time point in development and use them for regeneration. And these maps have been published and can be read. The first thing we did, we shoved these human fetal kidney stem cells in a model of chronic kidney disease. This was the remnant kidney model, and we could show that by transplanting these cells, again, this is old data, so I'm not going to go into it. You can influence the progression in mice, of course, of chronic disease turning into end-stage kidney disease. We can halt the progression by introducing, this was direct injections of human fetal kidney progenitor cells into the renal cortex of the remnant mouse model, where you chop off 5-6, you do a 5-6 nephrectomy, and then you do a contralateral unilateral nephrectomy. You shove these into the remnant model, and you can see that they can halt the progression of chronic kidney disease. So this was an example of how cell therapy can be relevant for renal regenerative medicines with the utilization of human fetal kidney progenitor cells. But life goes on, and if the people have been using and are using pluripotent stem cells, and this is not a protocol we've developed, but the protocol we are using. This protocol is, we have a BSF with Benjamin Friedman at WashU. So there are a couple of protocols where you change pluripotent stem cells into kidney organoids, and here you differentiate. There's a recipe to differentiate IPS or embryonic stem cells into nephron lineage, and get these kidney organoids in the plate, and this is how just how they form with Wnt agonist and media. There's a protocol you can use and generate the kidney organoids, form pluripotent stem cells, and you really don't need the human fetal kidney that we get in the lab. But let's see if you don't really need it. Remember that these IPS kidney organoids resemble fetal-like kidney tissue, okay? When you do transcriptomics of these organoids, they resemble about a 10-week human fetal kidney. So they're not maturing into adult phenotypes. You can find glomeruli, which are, are vascularized, usually minimally, and you need to transplant these organoids in order to get host-type vascularization. You need to get mouse vessels growing into these glomeruli, and you have some tubular structures in these organoids. So they're neat structures, but you have to understand they're immature, and they have also unwanted lineages which are non-renal. But you do see glomeruli and tubular systems in the IPS-derived kidney organoids. And what is very interesting, and this is data that is collected with Benjamin Friedman and Achia Orbach. Achia is from Bar Ilan, where this is a collaboration where we try to analyze the presence of the renal stem cells based on our biomarker system that we delineated in the human fetal kidney, and see if it's sustained in IPS-derived kidney organoids. And we have nice data to show that you can actually find this biomarker system preserved in IPS kidney organoids. And this would actually be beneficial, because then you could sort out your own early nephron stem cells and put them in a dish, because fetal kidneys, of course, are allogeneic. So you could do an IPS study and get the kidney organoids and use the biomarker system we discovered to isolate with ANCOM CD133 the early nephron stem cells, early developmental progenitors, and use them for autologous use. So the renal stem progenitor biomarker system we described is relevant also to IPS kidney organoids. But what about disease modeling? Now, everybody's using the IPS-derived kidney organoids for disease modeling. They have the human embryonic stem cells, they have the IPS, and we wanted to see what happens and compare if you use the human fetal kidney, not for regenerative medicine purposes, but for disease modeling. And this is Michael Namestikov in the lab. He's an excellent MD PhD student, getting human fetal kidney on a regular basis, getting this single cell suspension, and providing a protocol for growing human fetal kidney organoids. He needed to combine several known protocols by Belmonte, by Clevers, by different groups that have used these to generate adult kidney organoids or iPS derived. He has his champion media to get these human fetal kidney organoids growing in addition. This is how they look. They're complex, okay? And this is a P0, this is a P2. They can be grown for long periods of time. And we wanted to see what do they actually contain. They're ex vivo growing complex structures. Are there human fetal kidneys actually? So of course, the first thing you do is a transcriptomic assay, and you do bulk transcriptomics, and we found that these human fetal kidney organoids indeed express early nephron developmental markers. You can have, the kidney development is it's delineated. So you have these markers for each time point of development, for the early the early metanephric mesenchyme, these NPCs, the nephron progenitor cells, as I described. Later on, renal vesicle, S-shaped, different, again, more differentiated structures, and you can really plate these different markers and see where is your human fetal kidney situated, organoids are situated in terms of what do they comprised of. And interestingly, you can see that for instance, they're really enriched for early developmental markers, early epithelial markers. There are less, so they're less abundant with these NPCs, but much more abundant with renal vesicles and S-shaped comma figures. And this was interesting to see, because there are not many ex vivo systems that actually culture these cell types in high amounts. And you can see the GO analysis, renal system development, nephron epithelium development, renal tubule development, and more interestingly to us, notch signaling pathway. So notch is critical, I'll show you in a minute, for the growth of our human fetal kidney organoids. And we do a single cell analysis. They're not nowadays you have to see what cell types are contained with a single cell RNA-seq and pseudotime analysis to show that even though we NPCs are not abundant, we practically have, we have almost the entire composition of a human fetal kidney with some podocyte markers, NPCs, pretubular aggregates, renal vesicle here, proximal tube, distal tube, very neat cell types, which are, are maintained along the differentiation axis. But again, look at the notch signaling pathway, which is very abundant in these. This is again single cell analysis. You see each dot represents these notch activation in these specific cell types. You can see that very early on, notch is really activated. Once these NPCs differentiate to very early nephron structures, again, renal vesicle, S-shaped, comma shaped, you see notch being upregulated and activated. This is some stainings of the human fetal kidney organoids. This is, we need to compare to the in vivo. So this is human fetal kidney analysis, sections of human fetal kidney, and then you can really appreciate stainings once you have these human fetal kidney organoids stained. You can appreciate them when you compare them to human fetal kidney sections. And I'm not going to bore you with the different stainings, just tell you that they express markers of developmental markers of the renal vesicle, comma, and S-shaped type. This is the very early renal epithelium, just after the NPCs, the nephron progenitors, have generated the first, the most immature epithelial cell in the human developing kidney. So why use human fetal kidney organoids? You can use iPS kidney organoids, right? But look for instance, at the notch pathway or markers of renal vesicles. Here, please appreciate, this is the fetal kidney organoids. These are iPS-derived kidney organoids. This is LHX1 is a marker of the renal vesicle. Okay, notch activation markers of, again, renal vesicle, pretubular aggregates, renal vesicle, S-shaped, comma shape. Much, there's no comparison. The human fetal kidney organoids enrich much better these cell types than the iPS-derived kidney organoids, meaning that if you want to use about questions about cell types that are enriching human fetal kidney organoids, you might as well use them as opposed to iPS-derived kidney organoids. And again, this is a comparison between these two types of organoids. So I told you, human fetal organoids seem to be activating notch, so it is notch dependent, and we have notch inhibitors. Now, what's the relevance of notch to studying these organoids? Remember, there's a disease called Alagille. Alagille syndrome is a disease of many organs, but in kids they develop renal dysplasia, and this is an example of a renal developmental disease that can be studied in the dish. Alagille has mutations in JAG1 and activation in the notch pathway, right? So here you have the human fetal kidney in the Petri dish. You can block notch activation and see the phenotype that is developed and study its relevance to Alagille syndrome, a clinical syndrome that we treat and appreciate in the clinic. So this is a chemical inhibitor of notch. This is, you see the complex structures that are developing control organoids, human fetal kidney organoids, and once you implement the chemical inhibitor, you see these trouble structures. This is very nice. You can see the control kidney organoids here on the left, and here you see inhibited organoids. Again, these weird appearing troubled structures that I'm going to show you what they're made of, but crucially, notch is really obligatory for development of these human fetal kidney organoids. And you can see that LHX1 is reduced once we implement notch inhibition, and these LHX1 is expressed in the renal vesicle stage, and meaning that differentiation, once notch is not activated, differentiation in the kidney organoids will fail, and we'll get these again troubled structures, what we call, this is control, and this is the notch inhibited, what we call the emergence of troubled epithelial structures that are after, if they appear even after you cease notch inhibition. You culture these cells, you pass them to further on to a P2 culture, and the organoid is not formed, but rather these troubled structures, and we stain them. And interestingly, they're double positives, these structures, they're ECAD positive, EMA positive. These are markers of distal differentiation, while LTL is a marker of proximal differentiation. And what happens is that these structures are not separated. As I showed you, the renal vesicle and the S-shape need to separate in order to generate a proximal and distal tubule. But here, due to lack of notch activation, they fail to differentiate along the proximal distal axis, and we have these double positive troubled structures. So these are, I wanted to show you how Michael in the lab is studying a renal developmental disorder in order to understand notch activation, notch inhibition, and the relevance to a syndrome such as Alagille. But what about, how am I with time? But what about the adult kidney? I discussed the human fetal kidney sorting, getting progenitors, doing the regenerative medicine, and also disease modeling. But what's with the human fetal kidney? Now, again, I'm going to show you, this is the nephron and the generation of urine, as I showed you before, and getting urine here, and the shedding of cells. right? They fall all the time. And as I showed you before, there are 70,000 epithelial cells being shed into their urine, and we lose our kidneys into the urine. So we need all the time, as I said, we need to rebuild them. And each segment is rebuilt, as I showed you. I just wanted to repeat that. So we want to use these characteristics of adult kidney growth in our ex vivo systems. And what we do is we generate 3D kidney spheroids from the adult kidney. This is the adult kidney. The protocol was initially developed by Ella Boujor in the lab, and then was improved by Orit in the lab, which time Garari. And here you get the adult kidney, you get nephrectomies from the urologist. We call you up, we have a kidney coming out of the OR, and you can extract adult kidney cells, expand them, okay? You expand them in 2D. We need to get high cell numbers for regenerative medicine. We can't rely on small cell numbers. Now, these guys, the 2D expandable primary cultures, they differentiate. They undergo EMT as they expand. So we generate from these guys, from these 2Ds, we generate kidney spheroids, and this is how it looks. We have this special media. They aggregate in serum-free, and they generate these spheroids. Now, these kidney spheroids, you can analyze them for expression, and you can see that they express the different cell types or these unipotent progenitors required to build each segment of the adult kidney. As I showed you, there's not a multipotent stem cell in the adult kidney. So you can't have this 6-2 cell that will generate different types of the nephron. You need something that will generate the proximal tube and something that will generate the distal tube, and maybe even podocyte progenitors and different progenitors for different type segments of the adult kidney. So this is the spheroid that we generate on a regular basis in the lab. They are quite, once you generate, you need to expand large matrices of 2Ds, and then you generate these spheroids, because Ki67 goes down, and they're quite quiescent. You see, they're potent cells. They have a renal potency. They can generate renal tissue, but they once you generate them, you lose Ki67, you lose expansion ability, but you gain renal potency. And this is how it looks. Again, you take the adult kidney, you generate a 2D protocol, then the 3D kidney spheroids, and there you see the different colors here. Each cell type generates different kind of renal tubule. Proximal, the loop, the distal, you can get different kidney parts from these 3D kidney spheroids. Hence, there is a representation of these unipotent progenitors or the segment building cells and the kidney spheroids, and this is how it looks. Now, we wanted to even get better. We got these super spheroids, and what we do, and we want to get better expansions prior to generating spheroids, so we add an OCT4 to the adult kidney cells, and we generate OCT4 induced cells, and they can grow for many culture passages, and then we generate the OCT4 or supersphere, the OCT4 induced 3D kidney spheroids. They generate, they have a predication for the proximal tube, but they can be kept very, for long. They're very expandable. These guys are very expandable for many culture passages, and therefore they can be very relevant for regenerative medicine, where you need high cell numbers and expandable cell types. Again, different kidney parts by these super spheres, and this has not been published yet. But what about, again, using the human kidney spheroids to regenerative medicine? This is the low-hanging fruit. We want to see if it's comparable to the fetal kidney stem cells we previously isolated, and we used it again in the same disease model. This is the 5-6 nephrectomy disease model, where you take you do the 5-6, and then you do the contralateral nephrectomy, you get the spheroids. We do intrarenal transplantation. Okay, this is direct implantation into the remnant kidney. When you do a vascular infusion, these cells are retained within, they're stuck in the glomeruli, okay? They're not building tissues, okay? You can see them stuck in glomeruli. So you need vascular infusion is not relevant. You need to put them into the renal cortex. And once you put them into the renal cortex, you see massive differentiation, and you see self-organization and differentiation of renal forming of these renal forming cells into renal tubules. And what's interesting is once they form the tubule, they survive long term. Okay, cells when they're alone, they can't survive in host parenchyma. This is a hostile environment. So you get the renal tubules to engraft. You see massive replacement, massive engraftment by these self-organizing cells into renal tubules, and then they survive, and then they can exert their beneficial effects. So getting a structure is not only about regeneration, it's also about survival in host tissue and getting reasonable or good effects on the host environment. And we can derive these human kidney spheroid cells from normal kidneys, but also from end-stage kidneys. You can get end-stage kidneys that are taking out polycystic kidneys and so forth, and you can culture them, get the kidney organoids, implant them, and they do very good in transplantation assay. And again, this was published 2 years ago and covered and can be read. Again, some de novo tubulogenesis after transplantation and some cell replacement along within nephron epithelia, right? This is a chimera. This is a mouse tubule with human cell, mouse tubule with human cells. And these are this is de novo tubulogenesis, de novo generating new structures. And again, these tubules stay for a long time in a host parenchyma, and they can exert their beneficial effects. Again, higher magnification of engraftment. We have human markers that are very good to stain on the mouse background. Again, here they are. Human cell engraftment, human kidney spheroid-derived cells, self-organize in host parenchyma. Now, does it do anything to the renal function? It does. You can appreciate relative changes in blood creatinine or GFR. Now, remember, the disease is ongoing. The GFR is reduced, but it's reduced less in treated cells, and creatinine is elevated less in treated cells. It's not a complete repair. It's about halting the progression of disease and affording the patient more time until it reaches the end stage period. And this is what we call, this is a repeated measures model of relative change in blood creatinine, which allowed us to also draw GFR and show the functional effect in the 5-6 model. But how does it help? We generate these tubular structures, okay? We don't generate whole nephrons. There's no multipotent stem cells to generate whole nephrons in a disease model that a nephron would hook up would plumb into the system. We generate tubular structures and replace native tubule cells. But what we think is happening is we are reshaping the tubular interstitial compartment. Remember that CKD is, you have a glomeruli, but once you have kidney fibrosis and you have a tubular interstitial, a profibrotic tubular interstitial compartment, then you succumb to kidney fibrosis, which actually kills the kidney. Okay, it doesn't allow the kidney, even if it has a mere GFR, it suffocates the kidney. So this is the kidney spheroids. Actually, we call it reshaping the tubular interstitial compartment by generating of de novo kidney tubules, replacement of native tubule cells by fibrosis inhibition. I'll share that in a minute. These cell types exert. These cell types exert once they self-organize, they stay and they inhibit activation of myofibroblasts. there's the surrounding myofibroblasts. They inhibit collagen 1A2, 3A1, 4A1 in these resident cells. And what they also do, they release host tubuli for a G2M checkpoint inhibition, which, remember, senescent tubules which don't have the clonal expansion capabilities anymore succumb to CKD. Okay, so they increase proliferation and endogenous repair. So it's not only about generating new tubules and replacing native tubules, but also by fibrosis inhibition and really shaping the tubulointerstitial compartment, and by releasing G2M checkpoint and the inhibition in host tubuli. So these are what we call the therapeutic effects of kidney spheroids. And this is just a pathophysiology of CKD with senescent cells in tubuli and what we call myofibroblast and fibroblast activation. This is the type of accumulation of fibroblast, and this is a common final pathway of any of a CACUT, of a polycystic kidney disease, of glomerular disease. This generation of the tubulointerstitial compartment and I would say generating a profibrotic tubular compartment that is actually ongoing and injuring the tubulointerstitial compartment. And again, this processes influence GFR. So if you stop these processes, GFR can be retained for a longer time period. This is the scheme of transplanting human kidney spheroids into a mouse model, exertion of adult kidney cells from patient, expansion into generating the kidney spheroids, in vivo functional assays, human renal epithelial cell engraftment. It's epithelial cell therapy and the possible therapeutic effect, as I mentioned, generation of renal parenchyma, cell replacement, and also paracrine, autocrine effects. But again, they're done. These effects are done because these structures self-organize and are retained in host tissue, which is hostile, as opposed to other cell therapies, which cells come and go, they spill whatever they can, and they do not engraft. So there are also some putative connections that we can show between host tubuli to donor tubuli. What about a mix for regeneration? This is Oren's work in the lab, again published, where he said, and we said, let's do some more kidney form, not only kidney form stem cells, let's generate vessels together with renal parenchyma. So here we needed to use non-renal forming stem cells. What we used are to generate vessels. We used MSCs from bone marrow to generate pericytes, and endothelial progenitors were drawn from cord blood to generate the endothelial cells. It's interesting. I mean, these kidney forming stem cells don't generate vessels, and vessels forming stem cells don't generate kidneys. So you need to form tubules and blood vessels, you need to eject 3 types of stem cells. And this is the data: the MSCs from bone marrow, the endothelial progenitors from cord blood, the adult kidney spheroids, mixed with matrigel, subcutaneous injection, and intrarenal injection. And this is intrarenal injection. You can see the vessels generate fantastically. You do this MSC, EPC grafting. This is under the renal capsule. You see islands of blood vessels generating, and you can see cells within these vessels that are red blood cells. Okay, so they connect to the host vasculature. These donor-derived cell types connect to the host vasculature. These are CD31s. This is 14 days after graftment. And when you do intrarenal transplantation, you generate what we call renal vascular units from combined cell source. You generate peritubular capillaries, which are CD31 positive, human CD31 positive. You can see these peritubular capillaries together with human renal tubules, again, with generation of renal parenchyma. So by implementing 3 types of cells, you can generate renal vascular units, both renal lineage and vessels. Now remember, everything is lineage restricted, so renal forming stem cells generate renal parenchyma. Vessel forming stem cells generate both the pericyte and the endothelial cell. And this, again, this is a large vessel we could generate in the mouse kidney. This is an editorial in JSON where they showed, again, our work of generating host vessels that rejoin, donor vessels that rejoin grafted blood vessels with the recipient vessels, and generating a grafted tubule. Okay, so this is renal parenchyma again, and both and a blood vessel system, and they influence each other. And this would be the generation of renovascular units from 3 types of self sources. What about disease modeling with kidney spheroids? And here we jumped into the corona world, SARS-CoV-2. We had human kidney spheroids in the lab, and they express ACE2. We looked at it, they express ACE2. This is, of course, ACE2 is the receptor for the SARS-CoV-2. So we can use the virus with the Ness Ziona people, with the Biological Institute and Ness Ziona. We can infect kidney spheroids and 2D growing human kidney epithelial cells and study interactions between SARS-CoV-2 and the kidney. And this was published. And I have a funny story. I think that if we would have written, what happened is that the virus doesn't generate a cytopathic response, okay? We didn't see a profound, we have this VeroE6, the monkey kidney cell line. This was our control. You see this massive cytopathic response by SARS-CoV-2. But when you do the co-culture with the virus and you compare, you see that there is tubular injury, especially in the proliferating kidney cells, but we wrote that it doesn't generate a cytopathic response. And we think that there's no way in the world that the virus itself generates an acute tubular necrosis. And we think that it could join the wagon, it could help generating an acute tubular necrosis, an acute kidney injury, but this is not the primary reason for AKI in these patients. The patients are in intensive care, they have these, they have all complications of intensive care, and this is what they generate an AKI, an acute injury, and the virus can really deteriorate their kidneys. But so we were very, I would say we weren't showing the severity of the virus. And I think at that time the world wanted to hear about severity of the virus. So there were, the lack of cytopathic response was. We felt that the editors we wanted to hear a much more fibrotic response and so forth. But in general, speaking in general terms, the virus does infect the kidney. It enters the kidney. It could generate some tubular injury and you can see some tubular injury markers, especially in proliferating cells, less so in kidney spheroids, and it can also participate in acute kidney injury. And this is Dorit's Omer work in the lab. Last part of my talk is the urine. Leo, I need to finish. 5 minutes, you're on mute. 5 minutes. I just want to show you the wonders of urine, because we were dealing with the kidney cells, but now we practically moved to the urine. As I showed you, the 70,000 epithelial cells that are spilled into the urine can be used. And actually today we use urine samples from healthy and CKD patients. We follow the protocol we generated for the adult 3D kidney spheroids, but we did this. I didn't show the data here because we found some ways to culture these difficult to culture cells with expansion with specific inflammatory cytokines, and this is a pending patent. So we have the 2D expansion, and then you can generate the spheroids and use them for regenerative medicines and disease modeling, and sort of don't need the nephrectomy. You don't need a renal biopsy. This is what I'm aiming at. And again, this is just showing how urine-derived kidney spheroids are much better than 2D cells in terms of kidney epithelial markers, oxidative phosphorylation profile. They differentiate. They're potent. They look like kidney-forming stem cells. They're not the differentiated cells, and they can, again, just like the adult ones, the adult kidney spheroids, they can regrow them together with fibroblasts, and they can inhibit fibroblast activation. The urine-derived kidney spheroids. So they have this anti-fibrotic effect, which is relevant for kidney regenerative medicine. And this is the protocol Yehudit Gnatek does in the lab to generate, together with Doré, to generate these cellular models. We injected the cells, the urine-derived kidney spheroids, and again, self-organization and tubular structure. We use a different cell kidney disease model here, more sophisticated contralateral nephrectomy. We do a ischemia reperfusion and then a contralateral nephrectomy. But again, self-organization and an antifibrotic effect that can be appreciated by Masson trichome staining and analysis of Masson trichome staining in these kidneys. So the intradural administration of urine-derived kidney spheroids generates, self-organizes into tubular structures, and we see a reduction of kidney fibrosis. And these are spheroids. I want you to understand spheroids are led to aggregate, and they don't have polarity. It's a mean to generate a large amount of cell that are kidney forming cells just by doing the 2D expansion prior to and getting a high number of cells and generating the kidney spheroids. You can do a different experiment and generate the kidney organoids, which are you see polarity here. You can see polarity. The kidney spheroids are not polarity. They're these cell aggregates that can be used for transplantation or for I would say cultivating the using the viral interactions with the SARS-CoV-2. But these urine-derived kidney organoids, which we have the Israel Precision Medicine Program, again with Yair Anixter and Tomer Kaliski. We have the funding for generating these urine-derived kidney organoids and doing it personal. And this is, we've become a urine biobank, and we have these urine-derived tubuloids, for instance, of renal Fanconi. Okay, this is a Fanconi patient, and it's, Fanconi is a tubular disease, so it's a tubular adult disease. So it's really smart to use adult cells and use the urine because you have these mature tubuloids in the culture dish, and they can be passaged very slowly over many months. So you can see this is a half a year of protocol of having these urine-derived kidney tubuloids or renal Fanconis in the lab. And the kidney-derived organoids are not like spheroids. They have polarity. You can see a proximal tube ex vivo with LRP2. This is the megalin, this reuptake of albumin. And you can see ECA. This is a distal tube. In the specific localization, this is the luminal part of the proximal tube. This is the basolateral. So you can have the polarity, and you can use them for questions of disease modeling, such as renal Fanconi, the urine-derived tubuloids. And this is what we do. We generate them, we screen for transcriptomics, metabolomics, and then we aim to do high throughput drug screening to when we have the nice readout, a very robust readout, for instance, for these renal Fanconi syndromes, and of course, clinical assessment of the selected drugs. This is the precision medicine grant we have. So this is a comparison of adult kidney spheroids to adult kidneys, tubuloids. I'm gonna jump this. I'm gonna show you that each disease model can be used by a different cell type, and you need to ask yourself, what type of disease am I modeling? Am I modeling a developmental disease? Am I modeling an adult disease? Am I modeling a glomerular disease or a tubular disease? And each disease model has its own cells and its own protocol of cultivating a spheroid or an organoid. And I just want to show the people in the lab. This is just, Oren, the vascular work, Micha, the human fetal kidney organoids, Orit, the kidney spheroids. Ella started the kidney spheroid project. Dorit, of course, she's the orchestrator. The entire corona work was done. She pushed it ahead, and she manages both labs. And there's some people here, such as Asaf, Oren, which are, they're only they're PIs by themselves. So if we're not gonna bring therapeutics to the kidney, they probably will. So they're good progeny, and this is our support for lucky to have much support to especially now with the urine-derived tubuloids. And I call you all to collaborate on specific diseases that you have interests in, and we can use funding to find drugs and help our patients. Thank you.
Thank you very much, Benny, for a very extensive work you've been doing over the years. I haven't heard you the last couple of years, and it's nice to see that you are continuing at a rapid pace. While I'm waiting for questions, I have a couple of questions. First of all, regarding the disease modeling, your last slide actually described kind of what I was asking. In today's technologies, which types of disease do you think we can benefit the most from your models in terms of understanding mechanism and maybe, using it to identify either new therapeutic targets or optimizing patient-specific care? Can you model?
Yeah. I would say Leo, the low-hanging fruit is renal tubular diseases at the moment. The low-hanging fruit look. I would say this: the tubuloids can be modeled very nicely by the urine. The tubular diseases, Fanconi, Gittelman, Bartel, different types of tubulopathies along the nephron could be really, there are precise cell types that can be kept in culture, and UMOD nephropathy, for instance, MUC1 nephropathy. These are all tubular interstitial disease in specific segments of the kidney that are kept at ex vivo and can be amenable to RNA interference, can be amenable to drug screens. This would be the low-hanging fruit, I think. And so I think MUC1 has been screened by Anna Gerka at Harvard. People are moving towards that.
I see. And when you transplant, let's say, when you transplant these genetic disorders in vivo, do you get more information? Do you get more complex structures or something like this? Is it worth also, looking at in vivo disease modeling of these?
Yeah, I mean, look, yeah, you can do, you can get engraftment. Yeah, you can model them in vivo. You can get a renal tubule in vivo, but then it's very hard to screen, and I mean it's very hard to screen. It's very hard to screen these combinations once you engraft them. But I would say that this could be once you have your best target, right? Once you have your best target or your best drug candidate, you can engraft these complex structures and then see what happens in vivo. But I mean, to do the drug screen, blow up these libraries or do these antisense experiments, I would stay in vitro and then go in vivo once you have a excellent candidate.
And I was wondering, why is the IPS not as good as the human fetal kidney organoids, or is it just will not be good enough, or the field has not moved into maturing it or making it as good as?
So this is a great question, of course, and there are attempts by Ryoji Nishinakamura, the Japanese researcher. They want to push in renal stroma and renal vasculature, okay? Because once you implant the organoid, it matures, okay? Once you implant it, and it's done by host vasculature. So they're trying to implement different cell types in the kidney organoid. So I think maybe the new organoids can be, more mid-gestation human fetal kidney-like organoids. But it's interesting. We don't have, in our human fetal kidney organoids, because we don't have a lot of NPCs, we don't have what they have. They have very nice, they can generate filtration barriers, because, the glomeruli, they have nice amount of podocytes. We have the early nephron epithelia. Okay, so I think at the moment, if you want to analyze an HNF1 beta nephropathy, which is a renal developmental disease, or what we showed, Alagil, use our organoids. But if you want to do maybe look at glomerulogenesis and basement membrane defects, use the IPS-derived kidney organoid.
I see. Shlomi, do you want to say anything, or should we conclude?
Thank you, Benny. It was a very impressive presentation. We are running very late, so I will keep my questions to the next time we are meeting. Thank you very much.
Hopefully you can move to physical meetings soon, because today I think was the first day in Rambam that Cheder Ochel started again. So maybe we're moving in the right direction.
Coming to Haifa in the train is visiting you, Lior, would be of course great, and I want to thank you for the invitation. So
thank you very much.
Okay, thank you, Benny. Thank you, everybody.
Thank you, everybody. Bye bye.